Hello everyone, my name is Abhirama Ashira Jayaraman from DBT5B and my metric number is 23 DBT20F1035. The given topic for our presentation is Application of HPLC in Forensic Science. And my team members are Aishatul Afika, Noor Fitria, Sai Rajeshwari, Ami Ashraf and Hafiz. Those are the content of our presentation, which is type of sample, preparation of sample, suitable parameters, interpret the data, use evidence that shows this technique is the best method available for the analysis. Firstly, type of sample. We choose blood sample as our sample. Blood is a type of biological evidence which is commonly found at the scene of crime. Blood stains are often found at various types of crime scene such as homicide, hit and run, assault, child abuse, rape, robbery, and burglary. It also for DNA extraction established the paternity test. Blood is one of the most important biological traces that are often found at the crime scene. Due to the valuable information it contains, it is considered to be a very important forensic tool. Analysis of different aspects of blood stain can contribute to clarify circumstances under which some violent crimes have been committed. Such a crucial information can point at criminal investigation in the right direction and help solve the crime. It is very important to determine the sequence of events during the committed of a violent crime involving blood. Liquid chromatography has been used for many years for the separation, identification, and quantification of varied analyses. High performance liquid chromatography, which is HPLC, the advanced version of liquid chromatography, has been widely used as an analytical tool for research as well in industries. Forensic scientists, especially those who work in toxicology,
Hi, my name is Sarah and my matrix number is 23DBT20F1040. And now I'll be continuing the presentation by discussing on how to prepare the sample. So first, 2 ml of blood sample will be collected and then stored in EDTA vacuumainers. EDTA vacuumainers is a tube that is coated with EDTA. EDTA functions as it inhibits the coagulation process by eliminating the calcium in the blood. This will reduce the platelet activation and will protect the platelet when the blood comes into contact with the inner surface of the tube. Then the blood is analyzed in an automated cell counter to get the blood count. Later, the sample will be stored at 4 to 8 degrees Celsius. So when you want to run the sample in HPLC, 1 ml of plasma containing amlodipine will be added with 100 microliter of internal standard. Amlodipine is a calcium blocker, and internal standards are used to determine the concentration of other analytes by calculating the response factor. Then the sample is added with 100 microliter of 10% perchloric acid. Perchloric acid is to denature and to precipitate the protein in the sample. Then the sample is protected and later centrifuged at 4000 RFC for 15 minutes at 4 degrees Celsius. This is to separate the supernatant and the pellet. The pellet will contain all the proteins and calciums that we don't want. The supernatant is what we actually need. So the supernatant will be separated from the pellet. The supernatant is then filtered and then injected into the HPLC vials.
Assalamualaikum. My name is Ashata Afrika Akila binti Syamuddin. So I will be talking about the suitable parameters for blood testing in HPLC. So there are two analyses, which is the quantitative analysis and qualitative analysis. The quantitative analysis shows the retention time and the compound identification. Retention time is the difference in time between the point of injection and appearance of maximum peak. So the time takes for the specific compound to elute from the column after injection is known as the retention time. For compound identification, it is based on the chemical structure and molecular weight. Qualitative analysis is the measurement of the compound or concentration in the sample. It is used to determine the peak height and the peak area. So these are the factors that influence the performance of HPLC. First is the diameter size of the column chromatography, or known as the stationary phase. The small diameter of column chromatography will show higher sensitivity of the stationary phase. Next is pump pressure. High separation occur under high pressure. For sample size, it depends on the molecular weight and the chemical compound structure. For number 4 is polarity sample, solvent and the column. And number 5 is the temperature. The higher the temperature will conduct higher separation. So the sample extraction is potato glycoalkaloids, or known as PGAs, alpha-solanine and alpha-caconin. It can exert adverse effects on human health when consumed in excess. So the study aimed to investigate the optimal extraction method for the quantitative analysis of PGAs in whole blood by using high-performance liquid chromatography, or known as HPLC. So this validated the method was successfully applied to one forensic autopsy case, and the concentration of alpha-solanine and alpha-caconin in the postmortem cardiac blood. So these are the parameters to extract PGA from the blood sample. The type of phase that will be used is reverse phase. The mobile phase is 10 ammonium formate with 0.1% formic acid in water for solution A, and methanol HPLC grade for solution B. The column size is carbon 18 with diameters of 100 times 2.1 mm. The flow rate is 0.4 ml per minute, and the stationary temperature is 4 degrees Celsius.
Assalamualaikum, my name is Nur Fitria, and the next part that I will continue is interpret the data using the selected instrument, which is HPLC. So based on this method, the display wavelength that was used is 230 nanometer, and the flow rate was 1 milliliter per minute. During a 15-minute time interval was used. So after completion of the run, the pump was programmed to regain its initial conditions in 1 minute, and a 5-minute reconditioning time was allowed before the next injection. So the analysis of blood contents using a general purpose HPLC method confirm the results found in the preliminary screening. So in this method have two peak, which is peak 1 is paraquat peaks, and the second peak is internal standard. So the paraquat is a toxic chemical that is widely used as an herbicide, which is the plant killer, primarily for weed and grass control. So based on the chromatogram, peak 1, which is the paraquat peak, comes out first, then peak number 2, which is the internal standard. So the peak that comes out first is a component that has the same characteristic as the mobile phase, while the second peak, which is internal standard, is the component that is attracted to the stationary phase. So because of that, this peak is slow to come out of the first peak. So peak 1, which is paraquat, comes out at 5 minutes of the retention time, while peak 2, which is the internal standard, comes out at 8 minutes of the retention time. So the longer a peak comes out, the more efficient results we get. Based on this chromatogram, peak number 2, which is the internal standard, is more efficient than peak number 1. So that's all for my part. Thank you.
Okay, hi, Assalamualaikum. My name is Hafiz. My matrix number is 22 DBT 20F1045, and I will talk about the useless evidence that shows this technique is the best method available for the analysis. Okay, HPLC, a little bit of background about HPLC. Only compounds dissolved in solvent can be analyzed with HPLC. HPLC separates compound dissolved in a liquid sample and allows qualitative and quantitative analysis of what components and how much of each component are contained in the sample. Okay, why HPLC is the best in blood sample analysis? So in study 2001 by George Elizabeth et al., the aim of the study is to determine the cutoff level for hemoglobin when estimated by high liquid chromatography, which is HPLC, in the diagnosis of classical beta thalassemia trait. So this beta thalassemia trait is basically a blood disorder that reduces the production of hemoglobin. So they said that HPLC is a sensitive and precise method for the identification of HbA2, which is hemoglobin, HbF, and abnormal hemoglobins. It has become the method of choice for thalassemia screening because of its speed. For example, this cationic exchange column chromatography enables qualitative determination of HbA2, HbF, and abnormal hemoglobin in 6.5 minutes on hemolyzed set prepared from 5 ml of venous blood. Okay, the other reason why this HPLC is the best for analysis, for sample blood analysis, because of its reliability. So there's a compression on cellulose acetate, electrophoresis, and microcolumn chromatography. Level of HbA2 more than 3.4% is found in carriers of beta thalassemia. Unstable hemoglobin can be associated with an increase in HbA2, and in these conditions, the RBC index, which is red blood cell, that measure the size, shape, and quality of your red blood cells, may not fit in with classical beta thalassemia trait, while high performance liquid chromatography, which is HPLC, as a method for the measurement of HB, was rapid and technically easy. A cutoff level of HbA2 more than 4.0% predict the majority of carriers of classical beta thalassemia.
Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Amir Ashraf bin Ahmad Azam, and now I will explain the evidence that shows HPLC is the best method for blood sample analysis. Okay, the use of HPLC for measuring HbA1c is common in the laboratories of most hospitals, leading the authors to believe that this method could be used as requested by and under the supervision of a forensic medicine specialist. Should it not be possible to match the DNA profile identified in the blood strain recovered from the crime scene to a specific person, it will be rational to focus on people with a history of diabetes if HbA1c levels in the blood are found to be elevated. It will be appropriate to conduct further studies to determine whether the same results can be achieved in blood stains affected by different and particularly unfavorable conditions.